Hi everyone, it's Mr. Sinti, and today I'd like to talk to you about cell respiration. But here's the thing about cell respiration. It's a really interesting pathway, a metabolic pathway that occurs in all living cells. And what's interesting about this is that there's some complexity about it. And so what this video's intention is to discuss some of the biochemical pathways of it, or some of the chemical reactions of cell respiration that are important. And so, sort of like, if I can use an analogy, sort of like if I were a director or conductor of a symphony, I might want to, instead of just playing the song outright, if we were playing it for the first time, I might want to rehearse a few of the more challenging areas, because then we'll be able to play it fully. And so that's my intention here, is to get into a conversation in the basics of cell respiration, just kind of an overview, and also an emphasis on the chemical reactions of oxidation and reduction. Maybe this is a review based on some chemistry that you have in the background, but nevertheless, I hope you enjoy it and learn something from it. And so, one of the things I want to say right out of the gate is that life requires energy. And so there's a lot of chemical reactions in the cell that require us to produce energy. And where that energy is being produced, mostly in the cell, is in the mighty mitochondria. It's an organelle in eukaryotic cells. And so what's interesting about that is organic molecules, which are shown over here, could be often considered to be glucose, but it could be fatty acids. But organic molecules are combusted with oxygen, and that releases energy. It's an exergonic reaction, exergonic. So energy is released, and the products of these are simple, like carbon dioxide and water. And what's interesting here in this pathway, it's showing that in an organism that's photosynthetic, those organic molecules are produced by photosynthesis, like principally like glucose and oxygen are produced as a result of photosynthesis, and then the plant, for example, is able to burn the sugar and produce carbon dioxide and water, which is then in turn needed by the chloroplasts to produce the glucose. So it's a little cycle of life. But what's fascinating about this whole process, and this is where I want to focus, is that this conversation begins with the energy that's released from the combustion of organic molecules is used to produce ATP. And how ATP is produced is that ADP has a phosphate added to it. And then that ATP is then used to provide the currency to power cell chemical reactions that require energy. And so here's a picture of glucose here. And so cell respiration and fermentation are catabolic reactions that are going to be yielding energy, and that energy is then going to be used to produce ATP. And so where is that energy? That energy is stored in the chemical bonds. And so that's a little bit of an abstraction. So that's why we want to talk about this a little bit. And so as it turns out, enzymes, which are protein catalysts, will systematically degrade the glucose. It's not just in one step. And so it's a pathway, a rather long pathway. And some of the pathway is linear, some of it is cyclic. And it's kind of fascinating. And so my analogy here is that glucose, a source of energy that I think most people know has potential energy, is sort of like at the top of a staircase. Try to figure out what's the most known staircase in the world, and it might be the staircase found in the Titanic. And so glucose, which is up here, is going to sort of bounce down the stairs like this. It's going to go down step by step by step by step by step in pathway. So these are all chemical reactions releasing energy. So there's a release of energy when glucose is combusted with oxygen. But as it turns out, not going to go into the detail of this at the moment, but oxygen never really reacts with glucose specifically. There's a lot of steps. Oxygen comes in at the very end. And so all of this release of energy is used to take the molecule adenosine diphosphate and add the third phosphate to it to produce ATP. And so ATP is what we use in the cell for energy currency. So glucose, again up here, is reacting with oxygen. It's producing carbon dioxide and water, and then it releases a tremendous amount of energy, and that energy is used to produce ATP. And there's also a lot of waste in the form of heat, and so that's why there's a lot of energy inefficiency as if you were to relate this to the ecological pyramid of energy, and that's why 90% of the energy is lost when you move from one trophic level to the next, because it's a lot of energy is dissipated in the form of heat. So therefore, it's not available to the next energy source. So the combustion of glucose is exergonic, and all of that is used to produce the molecule ATP. And just to review, I would suggest watching the video on ATP, but just briefly. ATP is an adenine nucleotide connected to the ribose sugar in 3-phosphate. So collectively, it's a triphosphate. And so this is used in the cell to power chemical reactions. And so as I was saying a moment ago, food is burned with oxygen, and that energy, when it produces carbon dioxide and water, the energy from that catabolic reaction, cell respiration, is then used to add the phosphate to produce ATP, and then that is used to power chemical reactions in the cell. And so ATP is renewable. It's sort of like a battery that can be charged, and then it runs down, and then it's charged and it runs down. And so it's renewable. It's pretty cool. It's recycled in the cell. And so what are the chemical reactions of cell respiration? What are these chemical reactions? And what can we say about them in terms of characterizing them? What kind of chemical reactions are the ones capable of releasing a tremendous amount of energy that would be able to power the addition of a phosphate onto ADP to make ATP? And those chemical reactions that are most important in cell respiration are called redox reactions. And redox, as you may know from a background in your chemistry class, is a combination of oxidation and reduction. They're opposites of one another, and they happen simultaneously. And so one way to remember this is that redox reactions are ones in which electrons are moved closer to more electronegative atoms. Now you could try to remember that in terms of Leo the lion. So losing electrons is oxidation. That's what oxidation is all about. It's losing electrons, but the truth of it is it's not necessarily the total loss. And gaining electrons is reduction, or so Leo says Gur. So gaining electrons, but it's not necessarily the acquisition purely of electrons, but rather whether the electrons are being hogged a little bit more closer to an electronegative molecule. And I'll get into that in this particular video. So Leo says Gur, oxidation, reduction. And so these catabolic pathways in cell respiration relocate the electrons that are in a food source. Like, in other words, we're talking about like this, like an apple. We're going to relocate the electrons stored in glucose, and we're going to place those electrons into carbon dioxide and water. And in so doing, it's going to release a tremendous amount of energy, and that's going to be used to make ATP, and then ATP powers all the chemical reactions of the cell. So what's interesting, another way to remember Leo says Gur, so losing electrons is oxidation, gaining electrons is reduction. Another way of looking at this is something called an oil rig, like this. And so oil and then rig. So you can think of oxidation is losing electrons and reduction is gaining electrons. And so that's another way to remember this notion. So reactions that result in the transfer of electrons from one reactant to the other is a redox reaction. And so losing electrons is oxidation, gaining electrons is reduction. Okay? And so, when you're studying chemistry, basic chemical reactions, you may be familiar with this. And so let me start off with something simple. in inorganic chemistry, and then I'll sort of transition it to how we like to view redox reactions in biological pathways, like, for example, cell respiration. They're slightly different, but we're talking about the same thing, ironically. And so when you're talking about the formation of table salt here, what you're talking about is the metal sodium is reacting with the gas of chlorine. And so what happens here is that chlorine is a really electronegative atom. And so as a result, it's stealing away the electrons. And so there's a transfer of electron right there. And so there's a redox reaction. So sodium is losing electrons, chlorine is gaining the electrons. So here sodium is considered oxidized, losing electrons. Chlorine is considered to be reduced because it's gaining the electron. And so more generally, you could look at this in terms of X with an electron loses electrons to Y, which is then gaining them. So one way of interestingly discussing this is to say that, Y, or chlorine, Y actually caused the oxidation. Y caused the oxidation of X. Do you see that? Y is pulling the electrons away. So in so doing, Y is considered to be the oxidizing agent, like an insurance agent. It's the agent for which is gaining the electron. And so what's interesting about this is that the flip side of that is that X is considered to be the reducing agent because it reduces Y. Okay? So Y is the oxidizing agent. And in most cases, the oxidizing agent is, guess what? It's oxygen. But it doesn't have to be oxygen. It could be any of those electronegative elements which are in the upper right corner of your periodic table. But oxygen happens to be the great oxidizing agent in cell respiration. But there's other oxidizing agents, as we shall see. Okay? So redox reactions require, and it makes perfectly good sense. The reason it's called redox is if somebody's losing electrons, somebody's gaining them. So there requires both a donor and an acceptor. So another example, again, this one is also simplistic, but I'll go with it anyway. Like when you have hydrogen gas, okay, something like this, hydrogen gas, and you react it with oxygen gas. What is it going to make? Well, what it's going to make is a tremendous explosion. So unfortunately, this is what happened in the Hindenburg Zeppelin that exploded. Hydrogen gas reacted with oxygen, and a tremendous amount of heat was released. But the product of this chemical reaction is, of course, water, as you might be familiar with. A tremendous amount of heat energy is released in this redox reaction. And it's like, well, why would it be, excuse me on that, why would this be a redox reaction? Let's see if I can get this thing going again. Okay, so why is this a redox reaction? Sorry. Let's see, let's try that. Okay. All right. Let's do that again. Okay, so hydrogen gas, apologize for that. Sometimes stuff happens when I'm making a video and I have no idea. And so, okay, so heat is being released from this chemical reaction. So what gives with this? So why is this a redox reaction? Well, the thing about it is if you were to analyze closely, let me switch colors here. If you were to look at hydrogen gas like this, one of the things that it may be bothering you right now is that this is not a balanced chemical reaction. So I apologize for that. I was kind of thrown off by the technology glitch there. So as you can see now, it's a balanced chemical reaction. But nevertheless, do you notice here that this gas of hydrogen, the electrons are shared very evenly between these two hydrogens because the electronegativities are the same. So this is a nonpolar covalent bond. And so when you react that with oxygen, which is also shared very equally, so the electrons are going here. So when we talk about a redox reaction, sometimes taking it literally, like losing electrons, gaining electrons, it's really the elements that are becoming reduced. The elements that are becoming reduced are the ones that are holding on to the electrons more closely or hogging them. Like, for example, the product of this chemical reaction, oxygen and water. Check this out. When you look at the electrons here, again, they're being held together in terms of where they are very evenly. But as you may know, when this happens in water, the electrons are being hogged more on the oxygen side than on the hydrogen side. And so as it turns out, this is considered to be a partial charge of negative and a partial charge of positive. And so what I'm saying here is that oxygen is being reduced into water. And so what I mean by that is that the oxidation state, if you consider that it's not a true charge, and so if we say that it's gained like another electron here and it's gained another electron here, although that's not the truth because it actually is sharing it, but it's hanging out most of the time, we could say that the oxidation state of this is -2. You see that? And the oxidation state of this is +1. And so what's happening is there's a redox reaction, and in so doing, the movement of the electrons closer, here's the point, the movement of electrons closer to the more electronegative molecule is releasing energy. Okay? There's a tremendous amount of energy release as a result of a redox reaction. So losing electrons, that oxidation is causing a tremendous amount of energy release. So redox reactions can also occur, this is the point, when there's a movement of electrons that are closer in a covalent bond, like for example a polar covalent bond, but not necessarily a complete transfer. And so another example of this very point is that when you have, here's another classic reaction, when you're burning methane gas, it's also considered a redox reaction. And so what's happening with that? Well, as you know, when you burn methane gas, a tremendous amount of energy is released as well. But what's happening? Oxygen is acting as the oxidizing agent, and it's causing methane to be oxidized. And so what's happening? So here's the nonpolar covalent bonds right here, and here's the nonpolar covalent bonds. Now what's happening is there's a movement more toward oxygen here in carbon dioxide, and also a movement more towards the oxygen in water. So the oxygens are hogging, so there's a movement toward the oxygen, and so thus the molecule itself, carbon dioxide in water, has a hold, or it's hogging, gaining the electrons more than they were in methane and in oxygen gas. Okay, so here's an example of what we're talking about in terms of burning methane gas. Their energy is released. So when the bonds shift from the nonpolar to the polar, the electrons are moving sort of from a position that's equidistant between the nonpolar ones, and oxygen and methane, to more electronegative in the carbon dioxide and especially in the water. So oxygen is a real powerful oxidizing agent because it's able to pull electrons. So it's an oxidizing agent. So in other words, it's gathering and then gaining those electrons. And in so doing, oxygen becomes reduced into water. I hope that that's clear. And so the energy loss as it shifts to a more electronegative atom is a release of energy. Okay, so that's very important. The redox reaction relocates electrons closer to oxygen, therefore releases the chemical energy, allowing it to do work. And I know that that's still a little bit of an abstraction. So the electrons, if you will, and I put this in quote, because they fall. They don't always get transferred, but they're falling from organic molecules to oxygen ultimately during cell respiration, although oxygen's at the very bottom. And so there's some other oxidizing agents involved. And so cell respiration, glucose is principally, I like to discuss carbohydrate aerobic cellular respiration, but at another time we could discuss beta oxidation of fatty acids, and even amino acids can be used as a source of fuel. But basically, organic molecules can be broken down or oxidized into smaller molecules and releasing heat, and that's what we mean by redox reactions. And so the classic is glucose being burned by oxygen, producing carbon dioxide and water. So glucose is going to be oxidized, and oxygen is going to be reduced. Oxygen is the oxidizing agent. Although oxygen doesn't, and I'm sorry for being abstract about this, as we'll see in the next couple of videos on glycolysis and the citric acid cycle, sometimes called the Krebs cycle, and the electron transport system, we'll see that oxygen isn't really the oxidizing agent in glycolysis and the Krebs cycle, but it is in the electron transport system. But it's an oxidizing agent. It's going to strip electrons away from an organic molecule, and in so doing, those electrons fall more closer to the oxidizing agent, and so therefore energy is released. And so there's a lot more electrons in a triglyceride than in glucose. How much more? Twice as much. So triglycerides are a tremendous source of energy, potential energy. So hydrocarbons in general, and this is why we burn petroleum, and we burn methane, and we burn propane, and we burn octane, because there's a tremendous release of energy in those redox reactions. And so that energy sort of comes metaphorically down the staircase, releasing energy. And so these fuels, as it turns out, don't spontaneously combine with oxygen. And this is a kind of a subtle but important point. I mentioned it before. They don't react spontaneously. And when the word spontaneous can be misunderstood. Spontaneous sometimes refers to a chemical reaction that will occur, but slowly. And so what I mean by that is that it has a negative delta G free energy release. And in other words, it has a higher entropy, so it's positive S, and it also releases heat. So in other words, it has a negative H enthalpy. But enthalpy. So my point being is that because the fuels don't react quickly, it requires activation energy, and this is important because enzymes are therefore needed, and so therefore you can control how much energy is going to be pulled away from these electron sources. So in other words, long story short, is the cell can control when it wants to release the energy from when I'm eating an apple. And it can control it because it requires electrons to catalyze these redox reactions that are removing the electrons from glucose. And so they're removed rather slowly, but the enzymes can speed it up as necessary. So the fall of these electrons is usually captured by one of the most important oxidizing agents in cell respiration is a organic molecule which is a coenzyme, and it's an acronym as NAD+. Okay, and so most of these redox reactions occur during the electron transport chain, which is the third step in aerobic cell respiration. But I'm not going to get into it right now. I'm just going to get into a discussion of how NAD works in terms of redox reactions. So, as I was mentioning, cell respiration doesn't oxidize glucose in a single step. It actually takes many steps to pull all of the electrons away from those organic molecules. And so in biology, I mentioned that I was referring to the redox reaction producing salt, and I was talking about the redox reaction of the burning of methane gas. I was also talking about when hydrogen and oxygen gas get together to produce oxygen. But as it turns out, what we have in cell respiration is we have these organic molecules, okay, these hydrocarbons. I'll just make it sort of simple like this. Okay, so you get this carbon chain here with these hydrogens sticking off. And these tend to be, these bonds right here are nonpolar. And what's going to happen is you're going to get these electrons are going to be stolen away to something that's more electronegative. So this is going to be the oxidizing agent over here. The oxidizing agent is going to cause this to lose electrons. And in so doing, the way it's going to be losing electrons is that an enzyme is going to strip hydrogens away from the organic substrate and transfer it. And so let's get into that conversation. It's pretty cool. So glucose is the very first substrate in the pathway, but there's other fuels that are broken down gradually after that, and each catalyzed by a specific enzyme. And so, as it turns out, these hydrogen atoms are stripped, as it turns out, initially from glucose and other substrates, but they're passed to a coenzyme. Now, if you recall, a coenzyme is an organic helper that associates with a protein. So the protein being the enzyme, and the coenzyme being sort of a prosthetic, smaller molecule associated with it. And so this important coenzyme oxidizing agent in cell respiration is called nicotinamide adenine dinucleotide, or the acronym is NAD+. Okay, and so I know I'm sure somebody's shaking in their boots right now, but no fear. Don't be intimidated by biochemistry. We welcome it. And so this is actually a familiar, maybe looking structure to some of you. I mean, check this out. Here's our good friend adenine, which is a purine nitrogenous base, and it's connected to the ribose sugar. And then, hey, there's two phosphates right there. This looks like an adenine nucleotide, but it's a dinucleotide because there's two. And you're like, whoa. And then what's this up here? This is a nicotinamide. So it's nicotine with an adenine functional group over here. Okay, so this is what is the molecule NAD+. It has a positive charge. This is what NAD+ looks like. Almost through all of our discussion that's going to happen from now on, I'm not going to be showing you this structure because it's too huge. And so I'm just going to abbreviate this NAD+. You're like, well, what is this? This is a chemical that usually resides inside of a protein, okay? And it's like, well, what's it doing in there? What's it doing inside this enzyme? It's going to be an oxidizing agent. And if it's an oxidizing agent, it means that this molecule is very electronegative. In other words, it's going to really, really desire electrons. You're like, well, what's that have to do? Well, if it desires electrons, it's going to strip them away from food. It's going to strip it away from food, and therefore it's going to become itself, when it gains the electrons, it's going to become reduced. NAD+ will become reduced. And so let's take a look at this. And so there's an enzyme called Dehydrogenase, and it's a great name. Again, don't be freaking out. It's a great name because the enzyme itself strips substrates of 2 hydrogens at a time. So it's a dehydrogen, in other words, removing hydrogen, dehydrogenase enzyme. And it strips it away from food. Now, I'm specifically not using the word glucose because it's not always glucose. When it's broken down, it's something other than glucose. So we'll see this. But what happens is this represents, right here, this represents the substrate. And what's happening is this molecule, NAD+, is sitting in the active site. It's sitting in the active site of the enzyme dehydrogenase. This is the enzyme dehydrogenase. And what the enzyme is catalyzing is the oxidation of the substrate. And so, as it turns out, okay, here's the substrate. And it's like, well, what is a hydrogen? A hydrogen is a proton and electron, proton and electron. But what the enzyme is actually stripping from the substrate is this. It's removing, another color, it's removing one hydrogen and an additional electron. And so this is what's being removed, or loss. Loss of electrons is what? Oxidation. Oil, oxidation is losing electrons. And you're like, losing electrons? Yes, there's an additional electron right there. So what this is called is a hydride. It's a hydrogen with an additional electron. So hydrides are stripped from organic substrates and transferred to the oxidizing agent, which is NAD+. And in so doing, when NAD+ receives those additional electrons, it becomes reduced. And so as it turns out, it becomes NADH. NADH. And this is the reduced version of NAD+. And left over is an additional proton. Let's take a look at that. So here's the enzyme of a dehydrogenase. Okay, so this is a polypeptide. It's an enzyme. It's a catalyst. Here's some sort of food substrate. And the food substrate I was referring to before is having like hydrogens sticking off of it like this. Okay, so nonpolar bonds like this. And so what's going to happen? This enzyme, dehydrogenase, happens to have the coenzyme NAD+ sitting in here in the active site. And what the enzyme is going to do is it's going to strip these two hydrogens away. And what is it? Two hydrogens away are a hydride and a proton. And what's going to happen is the hydride is going to be picked up by NAD+. And it's going to reduce that to NADH. That's the reduced version. And so that's the redox reaction. The substrate is oxidized and NAD+ is reduced. And so let's take a look at that. Okay, so here we have the picture again of NAD+. It's nice to familiarize ourselves one more time. So here's the adenine dinucleotide. Here's the nicotinamide up here. This is the oxidized form of it. So check this out. When a hydride is acquired, in other words, it's picked up from food, so food is losing electrons. Who's getting oxidized? Food's getting oxidized. So what is it gaining? It's gaining, check it out up here, it's gaining the hydride. And when it gains the hydride over here, the hydrogen attaches to this upper area and the extra electron neutralizes that positive charge. So it becomes NAD+. None of this is affected down below. That's why we're highlighting just that nicotinamide up here. That's the part that's actually getting reduced. And who's doing this? The enzyme dehydrogenase is doing that, and then as a result, a proton is released as a result of that. And so the changes in oxidation form is from oxidized to reduced. So in cell respiration, we're going to see this often. We're going to see NAD+ become reduced. And in so doing, here's the most important thing. When it's reduced, it's now in possession of the electrons. It's now in possession of hogging the electrons more. And so therefore, it's a source of potential energy. NADH is now a high energy molecule because it has the electrons. And so it's gained the energy. It's gained it. And who lost the energy? The electrons had to come from somewhere. They came from food. So food is losing electrons and NAD+ is gaining them and storing them in the reduced coenzyme NADH. And I know what you might be thinking. You're like, all right, maybe, but what does that have to do with anything? You're like, what does NADH? Let me just go. What does NADH and now has all of this energy from food? Okay, let me just simplify it. Food is giving up its energy to NADH. So it's like now this has the potential energy. But like a waterfall, this is beautiful Yosemite Falls in Yosemite National Park. So what does this have to do with anything? We're going to see in cell respiration in a pathway called the electron transport chain inside the mitochondria. That's right. Inside the inner folds, right in here, there's going to be a pathway called the electron transport chain in the inner membrane called the cristae. And what's going to happen is NADH, which is inside the matrix of the mitochondria, is then going to give up. In other words, it's going to be oxidized. It's going to give up all of its electrons that it took from food. And in so doing, that release of electrons is going to power the production of ATP. Okay, no one said it was going to be easy, but it's totally cool. So the energy that's trapped in the reduced coenzyme NADH is going to be used to synthesize ATP as the electrons fall from NADH to oxygen in the electron transport chain, just as water falls from a waterfall. And then that's going to be used to produce ATP from ADP. Okay, so I hope that was remotely clear. And that's my video on the introduction to cell respiration and a brief look at redox reactions in a biological world. Thanks for watching.